Okay. So this, for under Lab 7, this is actually an old version of the chart that you guys filled out. I had to erase out some of the spots. But I'm going to go ahead and just turn it on because I always like, in case you guys lose your handouts, you can have it. So it's close enough to the real thing. So I probably should have just opened it up when you guys were working this morning. So again, it's the table of results, right, that you filled in this morning. It's just an older version. Because we didn't do all the antibiotics we did this time. So I had to erase away those not done's. So let me go back. So that's turned on for you guys. There were some videos to watch. Watch Peter Hickman do what you guys just did too, so that helps some of you guys to see it ahead of time, which is great, and I'm glad you did that. I had done a student version of this PowerPoint, so you could bring it to class because it has a lot of stuff in it. But of course it didn't have the answers to the homework, right? I removed those. So this one I'm turning on actually has all the answers in it for you. This is an older recorded lecture that I'm going to go ahead and turn on as well, because I'm not going to get through all of it. So if you want to watch the full version. Here's those practice problems you have for homework. So you have this, I gave it to you. This, on the other hand, is the answer key. I told you I would at the beginning of class. Here's me going over those problems step by step in a recording. So, Vanessa, if you need help or something, we can meet outside of class. I probably need to. This is that bonus quiz. So, again, you can print this out or just look at it on the screen and write your answers. Yeah, you can only take it one time. But, you know, you could write out your answers, y'all, on this sheet or on a separate sheet of paper, and you can come talk to me, even. And then submit it. No, it's online. Notice this is online. But I suggest that it just makes it easier to print out the paper version of it and write out and figure it all out. And then just click on this to enter your answers. So see, look, I'll open it for you so you see what it looks like. And so notice it says online. Yeah, this is just the questions. So here's the scenario, right? This is like what we did, the individual tubes. Oh, we didn't do the dilution factor, though, as we kept going. We will do that in this lecture, right? And then here's all the questions, right? And it's multiple guess, so that you can just fill it in on the quiz. So the next link is to the actual quiz, which I'm going to turn on later today. I'm not even going to turn it on now, so you can't accidentally click on it. Right? But notice it even has a link to the worksheet there. You may want to complete the worksheet before opening this quiz. So once you open it, it does have like the little sample thing in each block and then the question. So I mean, you could technically just do it online if you wanted to. I just find it's easier if you just work it out all ahead of time. Okay. So that isn't turned on yet because I don't want anybody accidentally clicking on it. I'll probably turn it on in like 2 days or so. Or if somebody emails me and says, I've done all the practice problems, and I've done the quiz, and I know I'm going to get them all right, so turn it on. Yeah, I don't think that's going to happen, even this week. But in a couple of days, I'll turn it on. Okay. All right. So today's lecture. I don't even know if I downloaded it. So there's a lot of information we're not going to quiz you on or test you on in this PowerPoint, and I really need to pare it down. So I'm going to try and give you the skinny. That's what I want to do. Because I have basically 45 minutes. Close your eyes. I can't do it. I was gonna like not be able to talk anymore. Okay. Alright, so for today's lab, we started out with antibiotic or antimicrobial susceptibility testing. We used a particular method though. There's a lot of different methods out there. We use the Kirby-Bauer method, or sometimes just referred to as the disc diffusion method, because we put little discs on, right? And what happened? The antibiotic diffused out into the media and affected the bacteria. And then we just set up a standard plate count, or sometimes what's referred to as a viable count. Viable meaning the organisms are what? Alive. So if we put any dead bacteria on that plate, are we going to see them next week? No, because dead bacteria don't grow. So we're only going to be counting what was actually alive in that culture that we started with today. And that's important, right? Because for the most part, we're usually only concerned about the ones that are alive, because those are the ones that can hurt us. So, you guys started out, right, measuring in millimeters the zones of inhibition on our plates, right? Except I wrote out lines for us to make it easier and so that they were as accurate as possible, because we didn't do it the next day. So in this picture, anyone know what type of organism this big white mass is? It's a mold. Specifically, penicillium, which produces an antibiotic we're familiar with known as penicillin. This stuff, streaked out on the rest of the plate, is a bacteria. Probably something like Staph epidermidis, a gram-positive bacteria. What did that mold do to that bacteria? It killed it, because that mold is actually producing penicillium. That mold penicillium is producing the antibiotic we know as penicillin. It's a chemical, right? It's diffusing into that media, and it actually kills the bacteria. Why are they fighting? Food and resources, right? Same reason we fight. And when we use antibiotics, y'all, we're just stealing microorganisms' technology. Their chemical warfare. We're taking it and using it for ourselves, right? We're growing these organisms in such a way that they produce the antibiotics. We take them from them and we put it in pill form and we swallow it. But there are some terms that people use inappropriately as it relates to antibiotics. Not all drugs that you take are truly an antibiotic by the strict definition of that term. An antibiotic is any natural antimicrobial agent produced by microorganisms. So again, it's stolen technology. There are drugs out there that are synthesized in a lab or made, say, by plants that don't come from microorganisms that will inhibit or kill other microorganisms. That general umbrella term that includes antibiotics is antimicrobials or antimicrobics. That's all substances, whether they're natural, synthetic, made in a lab, right, or from a plant. You're only truly using an antibiotic if it actually came from another microorganism and is affecting the microorganism you're attacking. So your safest bet is just to say antimicrobial, right? Because it encompasses everything. So the good news, too, is there are some natural and synthetic drugs out there that can affect viruses, that are antiviral in action. True antibiotics, though, are not antivirals. So microorganisms aren't making stuff that we're utilizing yet that affect viruses yet. At least the viruses that affect us. Right? We might discover it. Right? When we talk about antimicrobials or antibiotics, we put them in two main classes. Because in the definition, they either kill or inhibit, right? And specifically, we're usually trying to kill or inhibit bacteria. So bacterial, referring to the fact that we're affecting bacteria. Static, referring to that it doesn't actually kill them. It just inhibits their growth. Bactericidal, on the other hand, actually will kill them. So sometimes they'll be specific. Oh, this is a bacteriostatic antibiotic, or this is bactericidal. So for the ones that we use in this lab, I have them listed as to what category they fall into. Am I going to test you on that? No. What am I going to test you on? The definitions, right? The terms. All right. So how do antibiotics actually work? So for all the antibiotics we use in this lab, we know exactly how it is they affect either the growth or kill the microorganism. Am I going to make you memorize this for each one of these? No. I want you to have a general understanding of how these work, right? So in general, but for some of us, having a specific example helps, right? So I actually have, you like to know what you're working with, right? What it actually does. So that's on there. But I certainly don't want you memorizing it. So penicillin is the one most people know about, right? And the reason why this one works is because one of the targets that it affects is the cell wall synthesis. Actually being able to put the peptidoglycan pieces together to form the cell wall. When this antibiotic goes into the cell, it interferes with that cell's ability to make a strong peptidoglycan cell wall layer. Because of that, when the cells get ready to divide, because that wall is not strong and structurally sound, they burst. So what are other things that are really important to a cell in order to survive and grow? Well, they've got to maintain their membrane, right? Their membrane has to be intact. So if we mess with their membrane, could we inhibit them or kill them? Absolutely. And with cell walls, we can be really evil, right? Because guess what? We don't have cell walls. So if the drug affects the cell wall and doesn't affect us negatively, we can be what's referred to as being selective, right, in our toxicity. So other things we can target are the enzymes that replicate DNA. There are some antibiotics that interfere with going from DNA to RNA. What's that process called? Transcription. Should make Melissa answer it. From DNA to proteins, what's that process called? I know, because it's on your test. What's that process from RNA to proteins? What's that called? Yes, translation. So we can mess with the enzymes that cause that. Would that screw up a cell? Absolutely, right? They've got to be able to copy their DNA in order to copy themselves. They've got to be able to make DNA into RNA, and more importantly, RNA into proteins. And then they have metabolic processes that they have to do to stay alive, chemical reactions, right? So if we mess with those processes, right, the proteins that are involved in the metabolism or a particular metabolic pathway, we can inhibit or kill a microorganism. So as I said, for each one of these, right, it tells you what it actually does. So Cipro interferes with DNA replication. So it targets the enzymes specific to that method. So each one of them has a different target. So we saw something interesting today, didn't we? Penicillin didn't kill one of these guys, right? We specifically use staph because it's gram, anyone know? Positive. And we used E. coli because it's gram negative. E. coli is naturally resistant to penicillin. As you saw, it didn't get affected by the penicillin at all. Right? And the reason for that, anyone want to gander a guess on why it's protected? Because it has what? Why do you have your mouth covered? If you want to talk to me, though, you got to uncover your mouth. Oh, you're in pain. Okay. That would be a reason. Okay. All right. Anyone else want to answer? Okay, the cell walls are different. What else is different about these guys? Yeah, the peptidoglycan makes up the cell wall, so you have a very thick layer for gram-positives, thin for gram-negatives. Where do the antibiotics have to go? Inside, right? What do they have to go through to get inside a gram-negative cell? What layers? Outer membrane, cell wall, membrane. Gram-positives, cell wall, just membrane. This is an extra layer, right? Extra layer that antibiotic's got to get through. And guess what? Outer membranes don't like to let stuff in. They don't let penicillin in. So it's naturally resistant because of that outer membrane. Penicillin doesn't get in. Right? Selectively, that outer membrane's like, no, that's bad stuff. I ain't letting it in. No way. Ain't happening today. Right? So it doesn't affect them. So, again, just letting you know other targets, right? So, translation, cell wall synthesis, ampicillin. Wow, that sounds familiar. Sounds like another drug I just said, right? Penicillin, ampicillin. All the drugs that end in cillin, amoxicillin, ampicillin, these are all semi-synthetic antibiotics. They took penicillin and they modified part of that molecule to form this other drug. So in the case of ampicillin, right, it's a modified penicillin. Because of that modification, no, but amoxicillin's dirt cheap. That's like the liquid stuff you give your kid when they have ear infections, which is great because if you spill it, you can buy it without needing your insurance because it's so cheap. Because your insurance will only pay for it once. Been there, done that. Yeah, so it's a modified penicillin. So it's modified, I don't remember for that one, I think the action is it works against both gram-positive and gram-negative. Because they've modified it, so guess what? It goes inside gram-negatives. It's modified in such a way it kind of tricks them and they take it in. So you remember for that disc, for E. coli, for ampicillin, you measured it, didn't you? It was hopefully sensitive to ampicillin. We're going to use ampicillin later on in the semester. So this was an important one I wanted you guys to recognize, that ampicillin actually does work against E. coli because it's a modified penicillin. Does that make sense to you guys? Okay, so sulfur drugs that you may have heard of affect a particular metabolic pathway that bacteria have that we don't. Some people develop allergies to drugs, right? That's a completely separate situation and problem. So how many of you guys have this in your cabinet at home? Good old triple antibiotic ointment, or maybe you buy the expensive name brand Neosporin, right? No need. This triple antibiotic stuff is the exact same thing. Yeah. So guess why it's called triple antibiotic ointment? How many antibiotics does it have in it? 3. Imagine that, right? So if you read the label, it has polymyxin B, it has bacitracin, and it has neomycin. The problem with 2 of these is you do not want to take this internally, right? Although these drugs don't necessarily specifically target, but they can interfere with our cells' membranes and cellular processes, right? So if you actually read the label, right, you do not take this internally and you don't put it on large areas of your body because then you could absorb too much and affect your own cells. So minor cuts, right, and wounds. Oh, AD ointment is basically just Vaseline. I don't think it has anything in it antibiotic-wise. Yeah. It might have something else in it, but it's mostly Vaseline. AD ointment. Yeah. Yeah. So this stuff, notice the targets, right? We're hitting 3 different targets. What's the chance of some bacteria being on your skin that's resistant to all 3 of these antibiotics? Very slim. Because again, they're each hitting a different target. This one's affecting the membrane, this one's affecting cell wall synthesis, this one's affecting protein synthesis, translation. It's a 1, 2, 3 punch. We're serious about whatever is there. We don't want it to survive. Right? So, it's always important to completely read labels, right? And use things properly. So, small amounts on the skin, it's not going to be toxic. Large amounts is going to be a problem. And obviously don't take it internally. Oh yeah. It's the best stuff. So, resistance though is a problem, right? Because some bacteria, as we talked about with E. coli already, it's naturally resistant. The antibiotic doesn't even get in. So are there other ways they can be resistant? Yeah. So a drug's got to get in and usually interact with some target, right? Interfere with some enzyme or something that catalyzes the reactions for making cell walls or fixing membranes or making proteins or making DNA or some cellular processing. So it has a target. The problem is, for some of the bacteria out there, they've changed the target. So notice right here, that's what the target's going to look like the drug's supposed to bind. Down here, look, it's mutated. The target has changed. The antibiotic can't even bind to it. Doesn't even affect it. So that's one way the bacteria have changed and become resistant. Some of them have mechanisms like the E. coli, right, where it doesn't even go into the cell. They decrease the amount that gets into the cell. Some of them let it in, right, so it goes in and they're like, oh, no, that's bad stuff. Throw that out, right? So it comes into the cell and they throw it back out. They're like, oh, no, bad stuff. You cannot stay. And then the third thing that unfortunately a lot of bacteria have acquired nowadays are enzymes that digest antibiotics. So especially penicillin-like drugs, because penicillin-like drugs all have this basic structure. They all have this ring, what's referred to as a beta-lactam ring. And then this R group can be changed, right, to make the different cillins, ampicillin, moxicillin, all those guys. But this basic structure here, this ring structure, is in all the cillins. Some bacteria have made an enzyme known as beta-lactamase. It breaks this ring and deactivates any penicillin-like drugs. Which really sucks. Okay, so remember, molecules bond with each other, right? Amino acids bond together to form that peptide bond. That's partly where peptidoglycan gets its name, because it has some amino acids that bond with each other. So how does selection work? So we have a whole bunch of bacteria, right? And we apply an antibiotic. Are we going to kill all of them? No, we're going to kill most of them. But who's going to survive? The ones that are resistant. So over time, right, we're selecting for those resistance. And so what are we dealing with now? We're dealing with a lot of resistant bacteria because of the use and misuse of antibiotics over time. But they naturally are going to change and therefore potentially become resistant, even without the selective pressure of the antibiotics. But with that pressure, what we refer to as natural selection, we've increased the number of resistant bacteria. So there's a really cool video out there. It was an article put out by Harvard where they did this giant petri dish, right? And you can see evolution over time of them becoming resistant to the antibiotic. It's really kind of frightening. Okay. So that's the skinny on antibiotics, right, and how they work and why we study them. As I said, for each one of them I tell you exactly all the stuff about it. So how is it that some of them are resistant to chloramphenicol? Well, some of them have poor uptake, and some of them have inactivated the drug, right? So by no means memorizing, right? But like I said, we know for a lot of these what's going on. So zones of inhibition is where they're not growing, right? There's some terms related to that. We don't want to take a whole bunch of antibiotics, right? Because anything in excess, including water, will kill you, right, if you don't pee. So we want to use the minimum amount of the antibiotic that actually still kills the bacteria. So a lot of times we want to know what the minimum inhibitory concentration is. What's the least we can put in and we're still going to kill them? Another term you'll see is minimal bactericidal concentration. And this is the lowest concentration, again, cidal, that will actually kill that particular organism. So here, inhibit, right, we inhibit the growth. Here, we're actually going to kill them. Mean lethal dose, so when we're studying toxicology, so this is for anything that's toxic. So a toxin, radiation, or a pathogen. This tells us the dose that it would take to kill half of that population. Right, so when someone says the median, right, we're talking about the middle. So for some things, they'll calculate this out. Toxicity to us matters too, right? So what if in a Petri dish we can kill off, and actually one of the antibiotics we're going to use in a Petri dish later on in the semester, colistin, you would not take that internally. Because as it has, you can shut the door if you need to, an adverse effect on our own cells. So this can only be used outside of the body. We're pretty lucky with penicillin, right? It doesn't interfere with any of our stuff, and it's really specific in its target for bacteria. So we're being what's called selective, as I said, in our toxicity. We don't ever want to take drugs internally that would hurt our cells. Right? We want to be selective in that toxicity. So this is just a little bit different test than what we had. This is using strips, and the strips contain a concentration gradient that's measured out. So this is how much antibiotic is at this point of the strip. So 256, whatever their measurement is, micrograms or something. And where it intersects, right, is the minimum inhibitory concentration. So they could go as low as 0.04, and it'll still kill. Past that, notice it didn't, right? It grew right up to the end of the stick. So this is known as the E-test. Different from what we did, which was the disc diffusion. So how does it get from the disc into the agar? I just said it. It diffuses, right? It moves out through it. This is an important point. It moves out and down. Okay? So those plates that you guys used were a measured depth. If you were to measure them, they're 4 millimeters deep. This is to standardize the test. What happens if somebody screwed up? Which was the case. There was a few plates I didn't use because they were too thin. They didn't put enough agar in the tube for me. So it's not going to go as far down, right? So our zones are going to be, if we use those plates, bigger than they're supposed to be. Is that going to affect our results? Yeah, because how did you determine if it was susceptible or resistant? What did you do? You measured. So if your zone is bigger and it's not supposed to be bigger, is that going to affect your results? Absolutely, they're going to be inaccurate. The other thing, which for my other class wasn't necessarily the case, so this just tells you that I started with 250 microliters to make sure I got 4 millimeters deep. You could mathematically calculate it. Okay, the plates shouldn't have any visible moisture. Does moisture affect diffusion? So if I was to take a dry paper towel and a wet paper towel, and I put a drop of dye on the dry paper towel as opposed to the wet, which one's going to diffuse further? The wet. Because the moisture, right, is going to help that dye diffuse. So we don't want these plates too moist because what's going to happen? It's going to increase the diffusion rate, right? But if they're too dry, that could be a problem too. Right? So they have to be fresh. They need to not have too much moisture. So we use the spectrophotometer, right, to try and start with a standard amount of bacteria. And we used it right away, right? We didn't wait 30 minutes. I did those preparations right away. Why did we not wait? Why did I make them right away? What could happen to that bacteria in a matter of 30 minutes? So they're in the tube, they've got food, they could grow. Do we want them to grow? No, because we want around the same number of bacteria on all our plates, right? So you got to use it within a certain time frame. 30 minutes is the general rule. Anything past that, you pitch it and start over. So why did we pick E. coli and staph? Because one's gram-positive and one's gram-negative. And this does affect how antibiotics affect them because of their structural differences. So we already answered that. And then they had you actually do out a problem similar to what you did in class today, right? Where you had to measure. And the important thing is you have to look at that chart, right? To interpret. Do you have to memorize that chart, y'all? No, I will give it to you on the quiz. I will give you it again, right? You just have to interpret, right? So for this one, it measured 25 for chloramphenicol and 25 for penicillin. It's the same measurement, huh? But does it really matter? No, because you don't compare the measurements on the plate, right? You compare the measurement to each individual antibiotic to the table, right? So it doesn't matter that these are the same. What matters is that 25 for penicillin, that's too small, right? Penicillin's got to be 28 or bigger to be sensitive. In this case, it's resistant. Would you give that antibiotic to that patient with this infection? No. Can you give chloramphenicol? Sure, because that one has to be only 12 or greater. And it's 25, right? That zone's big enough. It worked. We could give that to our patient. Still with me? So what do you think you got to do for your quiz next time we come back to lab? Right? Or I'll give you the numbers like here. But you got to interpret it based on the table.
Check that against the chart.
Yep. And I'll give you the chart. Okay. So the last question, I kind of already answered earlier, right? How does the antibiotic get into the agar? Diffusion. But remember, out and down. That's why the depth of the plates matter. So the concentration as it diffuses, though, decreases, right? So what we're actually measuring is the minimum inhibitory concentration. Where some tests you would actually dilute out the antibiotic. Or in the case of the E-test, right, you would have that strip that has a dilution. What we're measuring when we're measuring our zones are actually the minimum inhibitory concentration. We're not actually getting the actual concentration, right? That's already been pre-worked out for these antibiotics discs. They have a specific amount of antibiotic on those discs impregnated into them. So again, you'd have to measure, right? And then interpret based on the chart, whether it was sensitive or resistant or intermediate. So you guys have already done that. You can see for Pseudomonas when we used to do it, it was resistant, like 0 zone, because again it's a gram-negative, so penicillin doesn't even touch it. But it was also resistant to the chloramphenicol. It was intermediate for the tetracycline, so you wouldn't want to use it. The only antibiotic that actually worked against that one when we did this was streptomycin. So this is just a table of the data that we've collected over different semesters, and should match up, it's just in graph form, match up with what you guys got today. Again, I'm not expecting you guys to memorize this, right? Just understand the importance and how to figure out whether it's sensitive or not. So our black bars are sensitive. Anything above the black bar is sensitive. Notice for some of these, right, the green bar is Pseudomonas. It only had like 2 measurable zones. And only one of them was sensitive. In this case, it was the Cipro when we tested against Cipro. And just barely against the streptomycin. The dark blue, right, is staph. As you can see, it was sensitive to all the antibiotics we used. E. coli was resistant to penicillin, right? So you don't see any E. coli band here. And where's the little blue? And vancomycin. So this is Peter Hickman, whose video you guys may have watched. He had a pretty scary bout with Staph aureus. So you can read about his story. So scientific method on what we did. Now enumeration of bacteria. So my 15-minute skinny on that. This is going to be interesting. So we could, right, take a sample, put it on a slide, and count how much bacteria. What do you think? Anybody going to volunteer for that? Sometimes, depending on how much bacteria you have, you would use a special slide that has a grid on it and a cover slip, and you insert a set volume into that square. And so since you know the volume and you can count microscopically, you can again calculate how many's in that original sample that you took a sample from. And for some things, especially eukaryotic cells, I used to do this type of thing. We unfortunately don't have any of these slides. Believe it or not, these are $400 a piece. But I'm hoping that we get some so that we can at least show you guys this technique. Can you tell if the bacteria are alive or dead when you do this? You can use a special stain that will help you know whether they were alive or dead. But once you stain them, guess what you do to them? You kill them. But again, you take a sample out of your original culture. Indirect counts can use things like biomass. We can measure products that they produce and kind of back calculate how many cells may be there based on what they produced, right? And machines can do that, or you can do what we did today. You can do serial dilutions and do a standard plate count. So this is very similar to what we did, except it's in milliliters and we did it in microliters. Same scheme though. Another thing that you can use, and that if I had time today, I would have actually read on the spectrophotometer the reading of the density of the culture that we started with. But I did not take the time to do that. If we did that data for each one of our tubes, we could plot that versus the number of cells we get on each plate and generate a standard curve for the organism that we use, so that you could actually use the spectrophotometer versus your data that you get on your plates. And in the future, you could just read it on the spectrophotometer. The problem with that is, again, with the spectrophotometer, it's not going to tell you whether they're alive or dead. Right? So it's going to give you a guesstimation of what you may have there. So in the past we would use these crappy devices and those long tubes, and you had to make sure you had the meniscus on the right line. But we've upgraded to what scientists actually use nowadays in the lab. We actually are using the micropipettors from biotech. So you guys got a chance to actually get your hands on them and use them properly. And I taught you how to set it for a P200 and a P1000. We didn't use the P20. But again, the settings are a little bit different. You got to know what it means, right, when you set it. And I think I have a video on that too. So counting is important, right? And so Peter has a little bit of back history on counting and that you can read through if you want. It's really interesting. And for some people it helps. Like I had to teach my son math not so long ago. And counting and all of that was difficult. And he actually really helped me out with this. If you think of zeros as being placeholders, right? Placeholders, it makes it so much easier, right, to count. So this goes through that. One important thing is the scientific notation. So we do need to be able to write our answers in scientific notation. You didn't need to be able to manipulate exponents. So if you don't remember that from math, or math is difficult for you, right now just go ahead and try and make an appointment with me over the next 2 weeks during office hours, right, to go through this stuff with you guys step by step. And I don't mind doing it. I love math. I'm one of those crazy people. Okay, so as I said earlier, we're going to flip, right, what most people do. We're going to flip our fraction. We're going to make it in lowest terms so that 1 is on the top, and we can just flip it. So the most part for the problems I gave you for homework, they're all straightforward, right? You don't have to do any major manipulating to fractions. I refer to fractions as evil Siamese twins because they're connected to each other, right? The top and the bottom, the numerator and denominator. And if you don't do the same thing to the top and the bottom, they get angry at you. In other words, you get the problem wrong, right? So that's probably my biggest tip when dealing with fractions is you've got to make sure you do the same thing to the top and the bottom. So, why do we do serial dilutions? Again, it wouldn't be practical to take that original tube and put that on a microscope slide and count each individual cell. Right? Depending on the volume you're dealing with too, it just would be too much. So if we dilute in series too, we dilute as we do each step, and we can reach higher dilutions faster than, say, if we put 1 ml into 1,000 milliliters of water to start to do in a dilution that we could do over series. And that was the part that I didn't draw out on the board. And I think I'm going to go back to the board and not use my PowerPoint at this point. So I have this recorded previously, and if this makes sense and helps you guys, watch it, right? What I'm going to try and do, and if you guys want, we can do the first example on the board real quick, and then we'll be done. So let me stop.